So this is to help you supplement what is going on at the moment. I've decided to record some of my slides for you just so that you have a bit more of a guided way to get through this whole section. So we started talking about cells already, and we did a whole bunch of osmosis, active transport, and diffusion. You saw that I added in some little bits and pieces for you guys so that you could actually do some revision on that. But yeah, now we're going to start exclusively with cells. So what I did include was a whole set of notes on this so that you guys have access to that. On top of it, you also have the slides. So what I recommend you do is draw where necessary, and I'll tell you what to draw, and take notes of what is important. And this also just serves to have a quick little introduction. So we are going to start with cells because I just think these things are the most ridiculous things on earth, and they're so tiny and they function so well. And in the greater scheme of things, cells are what makes up your entire body. So the average human being has approximately, I think it's like 7 trillion cells, and you shed these cells every single day to a large extent, and every single day, like approximate like 7 years, you actually are a brand new human being each time because you've actually shed all your old cells, and you've managed to regrow new ones through the process of mitosis, which is the next section of work. So let's begin. Okay, so there was a whole bunch of scientists, and you don't need to learn their names. They try to figure out, because they recognize that human bodies were really important, and they needed to know how they functioned. And they couldn't really explain how they functioned as well, for the simple fact that, they thought that you were just one big organism, and you weren't made out of these smaller building blocks. And this was a really important part of the world where eventually someone created something like a microscope, which you're seeing in front of you now, by Anton van Leeuwenhoek, who ground glass into lenses, and using compound microscopes, he could see bacteria, yeast cells, and life in a drop of water. So what you're recognizing here is that he's not seeing viruses, they're too small, but he's seeing very large bacteria as well as yeast, which is a fungus. So he's maybe some amoebas and stuff in the droplet of water. So he became, it was quite interesting. But you can imagine that through this, obviously the human race think that we far outdo everything else. So ultimately it needs to get a bit better. So then we had Robert Hooke, who was the first person to see cells, and he sliced like cork really, really, really thinly, and he saw that there were these box-like structures. And I recommend you go and Google this. He literally saw this black and white, and he noticed that there were these holes in certain places, but then he also noticed there were walls in other places. Then finally we had Lamarck. As Microscope improved, he announced that all living things were made up of cells. Can you imagine how absolutely scandalous this is? Now we're recognizing, oh my gosh, there's cells. Now what everyone used to think about, medicine has to ultimately change. All right, then we have Robert Brown, who noticed that every cell had a dark spot, and he called it the nucleus. If you go and Google the etymology of nucleus as well, you'll figure out that it means like the epicenter, or like where everything stems from. And this was very important because it was ultimately the nucleus, which was, very badly named the, what you call it of the cell? The, man, it's gone. The brain of the cell, and it really does function that way. Then we have Evangelista Purkinje, yes, who also worked on the heart. You'll learn about that next year. Cells consist of a jelly-like substance called protoplasm. So now we have this central organ, or organelle, and organelle literally directly translates into mini organ. And we know that it's suspended in this jelly-like substance. So you can imagine that if your cell was made out of pure liquid, every time you ran around, your nucleus would slide around inside of you. So you needed to have a slightly more sturdy substance that actually held the cells together. And this was really important because now we could recognize, because how on earth were these cells keeping us alive if they would just had this random dark spot and they would float around in this empty space? Then we had Schwann and Schlitten, which announced that the cell is the basic unit of life. And eventually by 1856, the cell's basic unit of life consists of a substance called protoplasm. Each cell has a nucleus, and each cells divide to make new cells. Now, before we continue as well, a quick little interesting fact. When one of the first microscopes were ever produced, and we actually got to look at cells, a lot of the time the microscopes were so bad that they weren't getting a true image of what cells looked like. So the churches back in that time were actually, well, they said that abortion was illegal for the simple fact that they found that when they looked under a microscope and looked at human cells, these human cells actually looked like a star. So it was this five-sided structure, and it looked like a head, two arms and legs. So they thought directly from conception or fertilization, the cell itself was a human, and they thought to themselves, like, it's murder if we actually get rid of that specific egg or that cell. So that's why they banned abortions at a very, 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 very young age without truly understanding the science behind it. Knott and Ernst Ruska invented the electron microscope. So now we're going really, really tiny. There are two different types of electron microscopes, and we'll talk about that much later. But you get the scanning electron microscope and you get the transmitting electron microscope, and I'll put some pictures up for you guys to have a look. Instead of light rays moving through electrons, small electrons move through, allowing greater magnification. So it's pretty much like you reading Braille, like taking your finger and moving it over those bumps to read whatever a blind person would read. The electrons technically do exactly the same. They run over the image and they bounce light back, or sorry, they bounce electrons back. And there's a screen that's able to pick up what it's looking at. And because electrons are so tiny, they're actually able to get really, really, really high quality images out of this whole thing. Also remember that electron microscopes are not something that we stumble across in a secondhand store. A scanning electron micrograph or a transmitting electron micrograph secondhand, and this was a couple of years ago, would go for about 12 million rand for one. And then you need an engineer to run the stuff as well. So it's not just something you can install in your bedroom and just start looking at stuff. It really requires a lot of space. But I mean, what is interesting for you to note over here, due to vacuum and dehydration process, no living cells could be viewed. So the idea is that, if I had any sort of air or water floating around in the channel where you put this whole thing in, we were going to affect the electrons quite badly. So we actually needed to put it under a vacuum and then dehydrate what we're looking at. So you might not be able to see living things and how they divide, but you're able to cut things up absolutely. So this is what I was talking about, the scanning electron microscope and the transmission electron microscope. The scanning, or the SEM, you can view in 3D, whereas the TEM, you can view sections of something. So you can actually cut a cell up like you would a tomato into very fine pieces or thin pieces, and you can actually have a look at it under the microscope. Okay, so cells are the basic unit of life. This is so important. So please note that you need to write this down. What do cells do? They release energy for cell activities, have the ability to produce new cells. They are mostly made up of protein, carbohydrates, lipids, nucleic acid, and water. And please note that those are your organic molecules. That's why I taught it to you in the previous section. Within each cell, tiny bodies called organelles are found, and in each organelle has a specific function. So let's go back to the top. So it releases energy for cell activities. If you did not have energy, you would not be able to function. So if you remember, the basic energy carrier is called adenosine triphosphate, or ATP. To a large extent, we need something to create that. Now, we'll go over what actually creates it later, but you guys do know what the powerhouse of the cell is, and the powerhouse of the cell is the mitochondria. So in other words, there is this little baby body or tiny body called organelles. So, to try and make something smaller, like in Afrikaans, you would go worm and then wormpies. It would be organ and organelles. So the L's part makes it really small. So inside is the mitochondria, and this thing causes us to create energy in the form of ATP. They have the ability to produce new cells. So you do know that each one of your cells has like a lifespan. And when it has reached that lifespan, it then gives up its job and it has to be killed. So a lot of the time, there's a protein called caspase or Cas9 or caspase 9, and it is able to indicate to a cell that you now need to commit suicide. And it will cause apoptosis. If you want to Google it, it's called self-cell suicide. I'm not even joking. And so when the cell's old and it's run its course, it actually just dies, and then a new cell is created afterwards through a process called mitosis, where you're creating genetically identical cells. All right. Then we have proteins, carbohydrates, lipids, nucleic acids, and water. I recommend you go and read the biological importances for all of these again, because now if you understand what protein is, you'll understand how enzymes are able to function within the cell itself. If you remember carbohydrates, you'll be able to recognize that it's used for the formation of energy. Lipids, we spoke about the cell membrane and how that's made out of phospholipids. So in other words, one of the organelles is actually called the cell membrane, and it's made out of these phospholipids, and I'll show you that later. And it's actually going to be quite like shock and horror to you guys when you recognize that your skin's actually not solid. It's actually made out of this liquid, and this liquid is technically called, well, phospholipids. Then we're going to have your nucleic acid, so we're going to have DNA and RNA. I recommend you go and like spruce up your deoxyribose nucleic acid and ribose nucleic acid. And then finally water. You remember I said to you, you need to learn like a minimum of 5 to 7 uses for water, because it's really going to be important. And one of the main ones you're going to recognize is that it is used for all chemical reactions. All right, so then we get all these organelles and how specific they are. So a lot of the time what you're going to recognize, and like the table I gave you for you guys to do from that PDF, is you'll recognize that it talks about how, the structure of them is very specific, and then we often relate its structure to its function. So like for instance, when we draw the nucleus a little bit later, I'll make you draw the whole thing and then get you to sort of specifically understand very specific parts. And if you understand what it looks like, you'll understand how it functions. All right, I think we're going to call it there for a day, and then we'll carry on in the next one.